Hello, my name is Maria. I'm a second-year postdoc fellow with the Schmidt Center. And let me introduce you Dr. Richard Bonneau. Richard's research focuses on developing, the Vice President of Machine Learning for Drug Discovery at Genentech Computational Science. Richard's research focuses on developing machine learning methods to power drug discovery. In particular, he is focused on biomolecular structure and structure-function relationships that underpin drug discovery. His work integrates structure biology approaches, bioinformatics, chemoinformatics, and natural language processing to derive new hybrid methods for design. Richard received a PhD from University of Washington. Prior to his role at Genentech, he was the director of New York University Center for Data Science. Richard, we're all excited for your talk, and the floor is yours.
Thanks very much for the invite and the introduction. Today I'm going to keep this a little bit high level, but we'll dive into just a few things around active learning and a few things around generation and sampling. But the first thing I want to say is that the title here, the Beyond the Funnel, the spirit of this talk is to sort of show you some of the efforts that we carry out at Genentech to integrate machine learning into drug discovery. And some of those things that we have to do are bookkeeping, some of them are end-to-end integration in the machine learning sense of the word, and others are process optimization. But the goal is to try to find the places where we take huge numbers of years and person hours of work, and we condense them to a single lead candidate, or we condense them to a single functional hypothesis, and therefore we make a place where if we fail, we don't really know how to go back. And so we're really going to talk today about end-to-end integration with the hope that at any given moment, we always have a large ensemble of leads, targets, hypotheses, so that we can be very fast and very parallel instead of sequential when we approach the drug discovery process. And I'm somewhat new to this. I'm part of the Genentech computational sciences unit. This was one of the first times where computational folks were elevated to VPs and SVPs at Genentech, and so I think this is part of Aviv Regev's vision to make computation a real part of drug discovery. I work with John Marioni in this new unit, Genentech Computation Sciences. So if you like genes, or cells or mechanisms, still reach out and talk to us if you're looking for things to do in this area. This is my unit. And again, I'll kind of go into where this fits in. We were Prescient Design. We were a startup company for about 7 minutes. And then I sent a note to Aviv, and the rest is history. We're now a unit or a department at Genentech that's divided in these ways. So some of the divisions here are somewhat canonical or old-fashioned, and that would be saying that we need someone to cover each of the two major modalities. And so atoms are atoms. All of the target genes and mechanisms are all made out of similar stuff in the human, but the way that we make drugs is still very strongly tied to two of the different major ways that we screen for hits and synthesize the material. And so even though we're very revolutionary, we still divide in this very classical way. The rest of how we divide things is to have a handful of folks who have no mandate. They just wander the halls in their bathrobes thinking about machine learning. This is led by Kyung-Young Cho, who is involved with the attention mechanism and involved with a lot of really cool work with Joshua in the beginning of this deep learning revolution. We also have folks that are focused on infrastructure, and we divide them into engineering and the large language model folks. Due to the size of these models, they end up thinking about engineering as much as they think about science. And so we kind of have the theory and the praxis, the application and the theory divided differently on different days. And in the end, we identify as scientists, and so we are always thinking about technology and platforms. But when we actually dive into the individual drug discovery projects, it's always quite a bit of fun. And if I use the word portfolio, it means individual targets that we're trying to drug. So this is a picture that was made mostly by Sarah Mostafavi, who's our new vice president of computational biology and translation. And I guess she's not that new. She's about a year old now. And she's 5 minutes older than I am at Genentech. But the idea here is that we have this inner loop, or this molecule-making part of the machinery. We have the target discovery and prioritization part of the machinery, but they use common infrastructure, common larger language models, which are the interface to this whole system, so that as we're figuring out what hypotheses, what targets, what pathways we think are important to modulate for a given disease, we are also in real time seeing what we could do to modulate those genes at the sort of composition of matter or molecule level. And again, we've got some older divisions and some newer, more technologically motivated divisions mixed in here. We're really excited about how we orchestrate this whole system with larger machine learning models. Today, I'll mostly give you a large molecule drug discovery focused introduction to what we do here. And the key message really is the utility of active learning, the utility of using models to collect the next sample. There's lots of other moving parts, but that's the vignette that I'll drop into in detail today, or in detail, I guess, talk detail. Before I start, I'll just kind of show this picture, which shows you the drugs or the medicines that have been approved and that are on the market, and that topped out in terms of sales. Now, again, we've got all sorts of things that we know about these drugs from our work as scholars, but also just reading the newspaper. And so there's a lot of different mechanisms here. There's a lot of really interesting biology behind these drugs. But what I want you to look at here is just how wildly different the composition of matter of all these things are. So we have peptides, but then they have some really far out chemistry to make them work, as far as clearance and the mechanism of the drug over time. We have antibodies, bispecific antibodies. We have pieces of viral capsids and coats. We've got RNAs that then express proteins and different things that are also very, very, very carefully engineered. And so there's a huge amount of bioengineering and chemical engineering, for the actual manufacture and process optimization of these. And there's a lot of molecular engineering that goes into all of these things. A lot of them are antibodies, but at Genentech, we're devoted to research along many lines across many modalities. And so for every modality you see on this slide, we have efforts at Genentech to optimize how machine learning can improve the design of those molecules. And again, we're going to drop into Lab in the Loop for antibody drug discovery or antibody-based drug discovery. When we say antibody, we still are doing lots of interesting things. We're making them pH and ATP sensitive. We're adding them into formats that have multiple valences and cross-linkers that have strange functions, transport mechanisms like the brain shuttle. But at some point, we do have to design the affinity part of that. And we have to do that in a context where we're paying attention to all those other properties. So these are the key ingredients, I think, for any recipe that designs antibodies well and that does so in a drug discovery context. And so the first thing to notice is that the key theme on this slide is this loop. And we can say without inventing any new math or without reading any NeurIPS papers or anything like that, that it makes sense to collect some data. And anytime you collect data, if you can, you should update your models. And some of that's engineering, some of that's moving data around. But once you have a model, what outputs from the model should you test? And so I encourage everyone to really think about active learning and model-driven experimental design as something that works substantially better today than it worked 2 years ago, and really, really, like quite a bit better than it worked 10 years ago. And there's a couple of different high-throughput biology, synthesis, the way that sort of CROs for making DNA and proteins are structured. There's a lot of things that are process optimized or just high throughput biology. There's a lot of other things that are also technologies that enable this that are on the machine learning side. But active learning to power this is key. So when we're doing this, when we're turning this cycle, we might be making libraries, we might be making plates, we might be doing things that actually have expensive tests that require animals, and there's a very small number of tests like that are allowable. But you should know how many tests you can make, and you should know the various elements of error in the model. tracking those things, you should figure out not just what to test, but how to diversify what you test. And then the key thing that people don't really think about that often is the bet size. How many things should you test given the time delays, the cost of a time delay, and the overall cost of each step here. And so that's something that can be done today. Obviously, we need deep predictive power for how we actually generate these antibodies, and we'll talk about that today. But I think the actual generative AI that produces de novo hits for proteins, we've all seen that, whether it's through media narrative, whether it's through reading the minutes of the Nobel Prize, or whether it's through actually following that field closely. I think this is something that we have a lot to contribute at Prescient, but this is part of the story that I think is told quite often. And so I'm going to talk less about that today and more about this. And then the last thing, these are sort of more controversial things that where I think the story is still developing. But at Genentech, we really believe that cross-modality thinking is key. And so whenever we can actually have a unit or team work on simulation for small, large molecule and peptides, cell therapies, we should do that as opposed to have them really drill down into the specifics of the screens or the modality that they're thinking about. This is something that I'll show a few examples of. And then the last thing, I'll end the talk with just a little bit of thoughts about how agents can be used to orchestrate workflows and perhaps allow some of this to run autonomously. But before we dive into that, I just want to highlight this key problem, or this key formulation of the active learning problem. There's lots of different ways to start thinking about active learning, but one of the ways to think of it is that with generative AI that's efficient at inference time, so you amortize your cost of using and building this model, you spend a huge amount of time to train the model, and then after that, there are lots of different ways you can sample diverse sets of things from that generative AI. And so you have a nearly limitless number of hypotheses about things that would work. Proteins, small molecules, synthetic biology, cell biology setups, we are usually in combinatorial spaces where the things that actually work, function, that hit the TCP for a given project, they're probably in the very large numbers of zeros regime. Once we have that core generation, we then have to apply lots and lots of filters. Can it be manufactured? Will it stay in the blood for a long time? Will it aggravate the immune system? That usually narrows things down, but we still have error in the inference of these multiple properties, and we're still starting on a, practically from a budget perspective, infinite field. How do we then select the subset that we want to submit for testing? And this is the core concept that we often use when we think about what are we going to do next in an experimental design setting, is this idea of Pareto frontiers. And here I'm showing a picture where we have, for example, two properties. One would be yield and one would be affinity. And in this case, these are real designs for an antibody that we care about. This is, I think, interleukin 6, which is something that it's approved for release. We can talk about it. I'll show you some data for that one. And so the things that we should test should do something novel or further from the origin with respect to both of these properties. And ideally, we should test the things that are at the frontier here. And this makes sense. And this can be difficult to compute when you've got lots of properties, but it's an under control problem. And this is slightly different from some of the other key principles behind experimental design, like de-optimality or block random. If you read a lot of early work in regression that might even be, might be so old that it's difficult to find, there's a lot of discussion about experimental design from models, but a lot of it assumes, IID, sort of independence of variables, and then a lot of it is also, furiously difficult to compute when you have a lot of data and a lot of dimensions. But another, really, really, really key thing that I think keeps us from using that ancient wisdom is just that everything that we do in living systems is really, really strongly dependent on each other, not just in terms of the variables having some correlation or some block diagonal structure or something that breaks the math. The bigger problem is just that they're dependent in a sort of a life or death fashion. And so, for example, if we have an antibody and I am very excited about all sorts of properties on its surface, but it doesn't fold or express. and I don't even know why it doesn't express. It just doesn't express. I don't get any yield. Without being able to make the thing, you certainly can't test anything else. And then if it doesn't bind the thing it's supposed to, Yan Wu, who's a VP in charge of antibody engineering at Genentech, she's very devoted to thinking about lab-in-the-loop and computation, but she is not going to be able to convince her scientists to do something with an antibody that doesn't bind, because from her perspective, it's dead, even if there might be immunological or surface properties that we want to test next. And so this is this little tree, but this tree is like a bush, and the bush is, from a mathematical perspective, burning. It's just a nightmare, because basically we get, millions of things here, hundreds of things here, off we go. And so if we can build Gen AI so that bigger fraction of things are binders, well, that's great, but we never avoid this key problem. And the paper that, I put this a little bit out of date here, I think the paper that I would recommend taking a peek at, and then reverse citation search will give you a lot of other things that Natasha has done, but this is Natasha Tavasovska's entry into this space with one of her good friends, Ji-Won Park. The two of them figured out ways to use Vine copulas and some other mathematical tricks to take the dependence tree of properties you might measure, and then use that tree to figure out ways to act like this, even though the reality is interdependent like that. And so that property DAG work and the paper Bowtied are two things that essentially let us then get together with our 60 closest friends in antibody engineering and actually try this active learning cycle to design antibodies. And this is the recent paper. The first author is Nathan Frey. The last author is Vladimir Grigorovich, who's one of co-founders of Prescient, along with myself and Kyung-Young Cho. This paper represents a whole lot of work, but basically for many different properties and for the 6 antibodies that we were allowed to talk about in this paper, because on the biology side, or for some other reason, partnership, they were no longer frontline projects. We were allowed to talk about 6 antibody design efforts here. We were allowed to talk about most of the properties that were involved in this, but this is essentially the pipeline that we've applied to all large molecule drug discovery projects across the Roche family. But here we're showing the results for a handful of projects where we had design rounds, and I think this is again interleukin-6. We had design round 1, 2, 3, 4, and if we believe this Pareto front or this active learning is working, we should see that our binding goes up and our expression goes up. And our binding here is in log of KD, and so as we go up here, we're getting to 10 to the minus 10 KD. We're getting to 100,000, et cetera, fold improvement in these seeds. These plots at the top are the distributions of therapeutic antibodies for a handful of properties computed in Charlotte Dean's Therapeutic Antibody Profiler, or the TAP program, which is at this point pretty much an industry standard for filtering things before testing. And then we also have BV ELISA here. This is Bacillus virus ELISA. So you basically rupture bacteria and see whether or not that mess interferes with the binding of your antibody. It's a really, it's a proxy essentially for clearance in the bloodstream. And so we showed that we stabilize these properties, and improve the 2 things that we tried to optimize against. And again, the key thing here is that over time, over a small number of designs, we improve binding by a factor of 100 for multiple antibodies, and we improve and diversify the set of antibodies substantially for all of these different targets. And these are a handful of different families that include well-behaved folds, membrane proteins, and other things like that. And again, I'm showing this one last time because active learning and model-driven experimental design is something I've cared about for my whole career, but it's not easy. It's really difficult. People graduate, people move to their next career phase, grants start and stop, and when you do things in biology, it doesn't take minutes or weeks, it takes years, and so it's difficult to have multiple rounds and really see whether or not your model-driven or automatically model-driven design works. And I don't think it works in every context, but for molecular design, it seems to be an ideal playground for experimenting with those ideas. Now, I said that I wouldn't talk a lot about automated de novo design of antibodies, but it is something that we have up and running, and we have integrated with library design. And the key thing here is to think through what it means to have the ability to paint an epitope and design antibodies. And so the first thing that this helps us do is to actually optimize and diversify antibodies. So a lot of times when we start projects, we almost always have a nanomolar binder to start with. And of course, we could compute these things computationally and then iterate, but Genentech is really good at making antibodies and has been for, I don't know, however many years, too many years to count, greater than 5, right? And 1 year is infinity in the machine learning field, right? So you could, of course, identify binders that bind different places. If you can use de novo, you can actually diversify not just the sequence, but you can actually rationally choose different regions around an epitope that you care about to try to come up with new functions. The way that antibody drugs and medicines work is often that you bind and you bring the complement pathway that's hanging out in the constant region and you induce cell killing, but that is one of 15 different ways that antibody drugs work. They work through avidity, bringing receptors together. They work targeting things. They work in just really numerous number of mechanisms. Once you get to multi-specific formats, playing around with the epitope and figuring out what's the exact geometry that optimizes the structure-function relationship important to the disease is super important. I think I just said complex formats, but one of the things that we care quite a bit about are the ability to engineer or rationally engineer things where you might have a targeting domain, a killing domain, a T-cell recruiting domain, or a receptor engaging domain. And so tri and quadra-specific formats are becoming actually somewhat routine. When we hand those off to development sciences and product development, they almost always have liabilities, and so the need to be substantially more active in designing complex formats is something that's important. The fact that this is fast, I guess, is the least exciting thing intellectually, but maybe the most exciting thing from the process optimization part. And then structure activity hypotheses here, this is the idea, again, with binder identification. If we're rational about the initial design, then we also probably have a good model of what's happening. All right, so that's another key thing. We need de novo hit finding, not just to avoid screening and immunizing animals, but to be rational and to be model-driven when we design. Another key thing is we have to figure out how to scale to lots and lots of different projects. And so here, this is, I'm switching gears to show you a slide from my small molecule group. And here we have this idea that we have a large number of things, including some things like potency modeling that we build in-house, active learning, which is this sort of overall sample selection. We also have things that we use open access code or we license code to do. But we have a large number of technologies that we have to apply to a large number of projects. And the right way to put that into an augmented design framework is something that we think about a lot. And so I won't go into that in detail today, but here we have issues of interface, issues of the active learning driving, but still allowing the chemist to override and other things like that. And this is something that's been really successful at Genentech and has led us to think about how far we can push it. So beyond interface, can we actually think about autonomous agents, and can we think about models that have been trained in such a way that they know the location and they have good vector stores for all the data at Roche. Roche is 125 years old, and that goes back to times where in Switzerland there were union laws, that whatever your mother tongue was, you were allowed to put that in your lab notebook. And so the lab notebooks in Roche are in a great number of languages. They're handwritten. They're 100 years old. So just from like a kind of a liking machine learning perspective, it's challenging and weird enough that it could be fun. But we need to build our own models for a number of reasons. One is that we have our own special data. It's not just the internet and a bunch of books. We also need to know that whatever we do, it can be compliant with regulatory processes. The EU just passed a whole bunch of laws about what can and can't be done with machine learning, and a lot of those are actually have specific clauses that target machine learning. And then a lot of the things that we do involve handing things off to clinical trials, and so there has to be some ability to put error estimates and safety estimates on these things, which I think you need to be able to at least see the parameters to do that. And so Kyung-Young Cho and Stephen Ra, who's formerly at Pfizer, put together a team to build our own large language models. This is something that's cross-Roche, cross-pillar, and is also being sponsored by Aviv and John Marioni. But in the beginning, we started off just saying, for whatever reason, we need to have models that are distilled to the smallest size possible that function in ways that commodity large language models work. And so we built large language models. We worked with large open access language models like the Llama Herd, and we found the sort of sweet spot for some of the tasks that were required from commercial down to research. The thing that we're excited about now, and the thing that I'll maybe take a beat to discuss a little bit, is this trend that I think is being adopted widely across different companies and different efforts to build these models. We want things that have deep expertise in vision and molecules. This is important for biology. This is important for molecular design. We want agents that are trained not just on the data, but also have deep tie-ins here, programmatic tie-ins. And then lastly, we want models that were trained and enriched either on the architecture or the training subset side to be good at reasoning. And we put these things together. We're really focused on tool calling in the design space. And our example or deep dive for reasoning is safety. So can you look at the literature and our internal data and make hypotheses about compound and safety? And here there's two things. Some of it's about toxicology, which has deep and ancient literature. Some of it's about on-target toxicity. So if you do the thing you do and it seems to be safe in a mouse, will it also be safe in a human? And the answer there is about, it's no, a third of the time, and it can be very surprising. And so there's a lot of work there in safety. For multimodality, we're really thinking a lot about the points where the green part, molecule design, touch the target discovery stuff that Sarah's groups are working on. We do have permission from the lawyers to release most of these models as open access models, and so this won't be top secret for much longer. But we do have some models that are trained on everything at Roche and other models that are trained with the intent to release, and you can imagine that makes everyone nervous. So we're working through that, and stay tuned if you're interested in playing with these models. Overall, one of our key takeaways here, and I'm showing you this somewhat older model here. This is updated a little bit, but this isn't our frontline model at this point. But if we look at models where we've pushed really hard to distill them down, they're very, very small. They take up a hundredth of the memory on the GPU that the larger public models take, or the larger enterprise models take. We're still able to, in a safety context, build competitive models without a huge amount of fine-tuning. In the context of tool calling, I think this is a case where just the last year of work has shown huge amounts of progress. These numbers, even a year ago, for even the biggest enterprise models, were below 50%. We see that we're able to get up to various correct response for the next tool, correct tool request. And again, the correct tool request is more about are the inputs and outputs compatible? This is about whether or not the user believes this. This is actually something where there's a Likert scale that the user uses to say, did this make sense? And then tool choice is, again, about not just compatibility, but more compatibility with the objective for the next phase in the workflow. And so we split these things up, but you can imagine the work to build these two plots was 70% metrics, 20% machine learning, and whatever's left, messing around with our high-performance computing just to get the darn model to train in the first place. When we put this all together, we do have this sort of user workflow tool interface combinations, and we've got something that's actually getting fairly full-featured for small and large molecules, and it's something that we're very excited about. And so this is a slide that's far from comprehensive, but it's showing a query and some ideas for what the model has sort of pre-packaged. Moving through that, we get questions about antibodies, we light up a few tools. We can use RAG to see what was used for that inference and what we might also look at. We can rank models and rank outputs, model outputs to test things. And we can queue up automation or experiments. And then augmented design is something that we love. It looks cool, it's fun to make, you get these cool interfaces, and then the chemists and the biologists don't really want it, but we're making it anyways because we're people too, we want to have some fun there. I think when it comes to these ideas of how we put all this together, I feel really, I wouldn't say comfortable, but I feel like I know what to try next on the active learning side. I feel like when it comes to agents, there's as many open questions as there are next steps. I think there's a lot of need to continue sharing our data layers and how we register models and encapsulate tools. There's a lot of work that still needs to be done there, or a lot of discussion. And then, the key issue for us is, if you're making a medicine, people have workflows, but those workflows aren't people who can make a medicine in a year. It takes too many years and too many people. And so do we embrace the long tail of all the different methods people use, or do we start to use these efforts to orchestrate things to also kind of clean up things and ask people to use fewer or a more standardized subset of tools? The answer to this might be that there's a popular answer, which is embrace the long tail, and there's a correct answer, which is ask people to change what they do, and we'll see where we get with that. And that's it. I'll stay on this slide for now and for questions.
Thanks. I think we have time for a couple of questions, but no more. So, okay.
Hi. Hello.
Okay.
Here,
Hi.
yes. Hello. Thank you. It's a very nice talk. So my question is, you have this lab in the loop, generate data and predict antibody and so on. So what's the relationship between the scale of the data set and the performance of antibody design? So how much data you need, basically?
Yeah, so there are a lot of extant antibodies and a lot of protein sequences. And so for the generative core, like the language model that suggests mutations or is, let's say, the proposal distribution generator, the underlying thing, I think right now we've seen that increasing the scale of those models, feeding them new protein sequences, is less important than how you balance, using ideas about evolution or the tree of life, how you balance what goes in there. When it comes to affinity and some of these properties, there are still things like clearance in the animal where we need proxy assays and we need more data. And then everything in between, affinity, expression, we're in a state where our models are very predictive. We think things work. But I think for the core unsupervised generative models, we're there. And for a lot of these other properties, we're actually working hard to collect new data. And so that's just the quantity. And we see that we could use a lot of additional data for many of the properties we care about. Not the core ones, not binding, not stability, not affinity. Now, the sub part of this question is how do you go about, for things like immunogenicity or clearance or high concentration behavior, how do you go about collecting new data? And there we have seen that the active learning motivated data collection is vastly more valuable than random or historical data. And so the data we've collected over the last 2 years is as valuable as the data from the last 10 years, for example.
Also a short question I noticed there's an anti-correlation between the yield and the affinity for the antibody design, right? There's anti-correlation.
I didn't notice that,
no,
but...
Okay. No, the higher affinity has a lower yield.
Oh, you're talking about that Pareto front picture that I showed? Yeah, I think that's one of those pictures where, my thesis advisor should have told me that there were too many points on that plot and that they were landing on each other.
See.
Generally speaking, we could make things a lot more stable, and we would. So you're right in one regard. There is that correlation. The most stable and the best expressing proteins are very unlikely to be the best binders, and the best binders could involve all sorts of hydrophobic and large loops that would then be problems for those other things. And so I think that anti-correlation for the extremes of those properties is probably, probably something we'd observe. And now I don't know also if that's also a trivial geometric result of how you draw Pareto fronts. So we should think about that a little bit too. Yeah, that's a good question.
Okay, let's thank the speaker again for the amazing talk.